In addition to the HLA class 1 genes, the human leukocyte antigen 1 genes in the region of chromosome 6p21, there is a neighboring HLA class 2 set of genes. This can be subdivided, this region, into a DP region with 2 genes, functional genes, there are additional pseudogenes, a DQ region, and a DR region. These 3 regions then code for 3 types of MHC proteins, which have both an alpha and beta unit. And so the DPA1 and DPB1 genes code for proteins which then form a dimer. And note they are very variable, with 39 alleles and 520 alleles respectively. The DQ genes have 54 alleles and 780 alleles, once again very polymorphic and forming a complex together. And then while the DRA1 gene is not very dimorphic with 7 alleles, the DRB1 gene is one of the most polymorphic genes known with over 1,700 alleles. Since these genes are located closely together, someone who inherits one specific allele is likely to inherit the allele of the neighboring gene. And so therefore, very often alleles travel in blocks known as a haplotype. So even though they are separate genes, they travel almost as if they are one. And there can be linkage disequilibrium in that 2 genes are associated together more frequently than one would expect, suggesting that selection is keeping specific alleles together. Some alleles affect disease vulnerability. In the case of diabetes, there are alleles which offer protection from diabetes, given that diabetics rarely possess these alleles. There are 2 alleles which are associated with 90% of those who have type 1 diabetes having either one or the other allele. These alleles can also occur in non-diabetics, but to a lesser frequency. And 30% of diabetics possess both these alleles, the DR3, DR4 heterozygotes. These genes are those which offer the greatest genetic link to type 1 diabetes. These HLA2 genes form the proteins known as the MHC2 complex, which instead of being found on every nucleated body cell and presenting the peptides which originate inside these cells, these MHC2 proteins are only found on antigen-presenting cells, those cells which take the foreign peptides which have undergone phagocytosis and then present them to immune cells, the T helper cells, to stimulate immune responses. Cells which are antigen-presenting cells include macrophages, dendritic cells, B cells, and a few others to a certain degree, such as epithelial cells of the thymus during development. After phagocytosis of an antigen, these MHC2 proteins bind to a peptide once the proteins of the antigen are broken into fragments, and then carry this peptide to the cell membrane where this APC can then present to a T helper cell, a CD4 T lymphocyte, and the APC will typically then stimulate it with a local hormone, and then the T helper cell then proceeds to stimulate additional immune responses.